Hi everyone, I'm sorry I couldn't make it into class today or on Friday. I'm afraid I was unwell and wasn't able to come in. I'm glad you got the test done anyways, and I'll be marking that this weekend. So I want to move forward with what we have left in the semester. So we've left the anatomy portions. Now we're into a little more of looking at the muscles and some physiology in regards to contraction and so forth. So this first slide, or this first group of slides, is Introduction to Muscles Part 3. We've already discussed muscles and looked at, why they're named and the sizes and so on and so forth. This has to do with the microscopic structure of muscles itself. So we're going to look deep down into the muscles as they look when we look under a microscope. As we've talked about before, I've alluded to the fact of this connective tissue covering we see on muscles. I've drawn some pictures in the classroom that show where a muscle is with the extended tendons out each end, and I've also talked about how when the muscle fibers shorten, they produce tension. That tension then affects the internal connective tissue. And because that internal connective tissue is fully connected all the way out to the external parts of connective tissue of the muscle, it too then extends out into the tendons. So the shortening creates tension, and that tension moves all the way out to the tendons and its attachments. And therefore, we get muscle shortening and movement as it relates to the joint that the muscle crosses. So as we move down into the deeper parts of it, we say that each individual muscle is separated and held in position by layers of fibrous connective tissue called fascia. And you've heard me use that term. I'm sure you've heard it in the lab a number of times. Fascia, fibrous connective tissue, is sometimes called deep fascia when we look at bones and muscles. And as I've said, at times this connective tissue, it projects beyond the bulk of the muscle fibers to become what we know as the tendon. And the fibers of the tendon intertwine with the fibers of the periosteum, therefore attaching the muscle to the bone. So it isn't that the tendon or the muscle and its tendon are directly attached to bone itself. It does have this sharing of tissue between the fibrous parts of the tendon and the parts of the periosteum. These intertwine, which makes a very strong connection. And because of this sharing, we say that that's where the muscle attaches. As we've talked about before, this same connective tissue can form broad fibrous sheets that can form for attachments, such as the thoracolumbar fascia. And this, as we know, is called an aponeurosis. And this can allow for attaching a group of muscles or adjacent muscles. The connective tissue that surrounds the outer part of the muscle itself and all the connective tissue prevents the muscle generally from bursting when there is a strong contraction, and does allow for a significant increase in tension, which of course ends up in movement. The connective tissue also holds the fibers in a parallel relationship. So when we look at the muscle fibers themselves, they run parallel along the, shall we say, longitudinally between the two attachments. So if we consider where a muscle attaches in its 2 points, which many muscles we've talked about finding attachments, the fibers run parallel to each other from one tendon to the next. Now, we did talk about the various fascicular patterns. Remember the pennate muscles, that kind of stuff. So, but they still run parallel to each other. So even when they spread out or they have a particular shape to them, the direct muscle fibers themselves, when we look down in at the muscles, are still running parallel to each other. So we can see here, the drawing shows the tendon here attaching to the periosteum. And we can see the fibers here running parallel from that tendon to the next. We can also see within the packages here, the fibers again are all running down parallel to each other. And we're going to talk about all these different layers of connective tissue. So as we said, skeletal muscles are wrapped in connective tissue, and together this wrap is continuous with the deeper layers. So remember, the reason we name a muscle is when we look at the outer connective tissue layers, we can separate one muscle from another, and hence the reason why we talk about one muscle versus another. But that connective tissue that we see on the outside of the muscle runs deep into the muscle itself, all the way down into the deep parts of the muscle. So we say that it's continuous with the deeper layers. So that when a muscle is contracting, it pulls on the sheaths of connective tissue that surround it, which in turn transmit the pulling force on the bone to be moved. So all this connective tissue, as I said, from the most superficial parts of the muscle, runs very deep into the internal parts of the muscle, and is also the same tissue or the same connective tissue that creates the tendons. So therefore, when the muscle pulls on all this connective tissue, that force transmits out to the tendons, which results in the bones moving at the joints. The connective tissue layer that surrounds a muscle superficially is called the epimysium. And it is a coarse sheath that envelopes the entire muscle. And as I said, this is how we individualize one muscle versus another, because we can see this outer coating that separates, even though they may be very intimately close to each other within under the skin within the area of the body we're looking at, we can individualize them because they have their own specific epimysiums. From the epimysium, this connective tissue extends inward and separates the muscle tissue into bundles of cells or bundles of muscle fibers that are called fascicles. And these are bundles. So we can see here that we have these bundles here. So we have the outer coating here in blue, which is the epimysium, which is the fascia that surrounds the outside of the muscle. And then it in turn starts to go deep into the muscle, and it separates bundles of fibers. And these bundles that are surrounded by connective tissue are called fascicles or fasciculi. So we can see that they're surrounded here, and we can say that, that this epimysium does go in and create these bundles called fascicles. The connective tissue that surrounds the fascicles, even though it's a continuation of the epimysium, when it surrounds the fascicles, this is known as the perimysium. So the perimysium surrounds the fascicles. The perimysium then invaginates or travels deeper yet to surround each individual muscle fiber. And the connective tissue that surrounds each individual muscle fiber is called the endomysium. So the epimysium surrounds the whole muscle superficially. Then it invaginates deeper into the muscle, where that connective tissue then creates packages or groups of muscle fibers or bundles, and that is known as the perimysium. And then the perimysium travels even deeper still to surround each muscle fiber, which is called the endomysium. Now, because all three of these structures are continuous with the fibrous structures that attach the muscle to bone, the muscles are firmly harnessed to the structures that they pull on, which, as I said, results in that any tension that's created in the fibers themselves extends out into all three of these layers of connective tissue, which then of course extend out to create the same connective tissue, to create the tendons and its tendinous attachments. So any tension created in the muscle itself extends out to create tension within all the connective tissue layers of the muscle, and therefore also the associated tendons. This type of a configuration of this connective tissue allows for independent movement, and it also allows for things such as blood vessels to exit or enter the muscle and for nerves to pass through. So we can see here in blue, the outside of the muscle is surrounded by the epimysium, the fascia of the muscle. It then extends inward to surround bundles of muscle fibers known as fasciculi, and this connective tissue that surrounds the bundles is known as the endomysium. And then each individual muscle fiber is surrounded by its own, the perimysium surrounds the bundles, my apologies. So epimysium surrounds the muscle itself. The perimysium creates the muscle bundles, and those bundles are known as fasciculi or fascicles. And then each individual fiber is surrounded by connective tissue that extended deeper from the perimysium to form the endomysium, which surrounds each muscle fiber. The other thing that you can see here is that, within those individual fibers, even though they are surrounded by connective tissue, we can see how blood vessels have come in. And of course, there would be associated nerve fibers there as well. We've mentioned this before, that when we look at a muscle cell, we call it a muscle fiber, but more importantly, we either call it a myofiber or a myofibril. So whenever you see a myofiber or myofibril, that is associated to the fiber of the muscle, but it is in fact actually a muscle cell. And they're called fibers because they look like a thread or a hair, hence the reason we call it. So when we think of it that way, we can then say a muscle fiber is an elongated cylinder which can extend the full length of the muscle. So if you think of your quadriceps, that originates, you have a muscle, you have a tendinous attachment up at the AIIS of the pelvic bone, and then you also have a tendon that goes down to the patella that would then extend down to the tibia. So between the tendinous material to the patella and the tendinous attachment to the AIIS, you have muscle fibers that extend that entire length. So if you just look at your quads, that is a considerable length for a fiber from a, should we say, from a microscopic perspective. That's a relatively long distance. And picture that you have, the quadriceps being individualized into its 4 parts via the epimysium. Then you have the fascicles running in there, the perimysium, and then each fiber surrounded by the endomysium, running that entire length from the pelvic bone all the way down to the patella. It's fairly long. So we can see here again in this drawing, here we have the perimysium, of course, that's going to surround the outer part of the epimysium is going to surround the entire muscle itself. We then have the perimysium that is surrounding the bundles or the fascicles of fibers themselves. And then we have the endomysium here that's surrounding each individual muscle fiber that's heading outwardly. When we look at a muscle fiber, the muscle fiber is made of sections known as sarcomeres. So those sarcomeres are the working part. They then are within a cell and they are surrounded by a cell membrane. Now, so if you think of a myofiber as being a cell, it has to have a lot of the normal things we see within a cell, of course, one being the cell membrane. And when we look at muscle cells, the membrane of a muscle cell is called the sarcolemma. And again, when we look at the extent of length, right, of a quadricep, that cell muscle membrane can be as long as 30 centimeters. So we have the sarcolemma that's the membrane, and then within the sarcolemma, of course, we have the cytoplasmic material and so forth. Well, within muscle cells, this is known as the sarcoplasm. So the sarcolemma is a cell membrane, and then the cytoplasm is called the sarcoplasm when we look at muscle cells. Within that sarcoplasm, you have large amounts of glycosomes, which are stored glycogen. And if you remember from our glucose metabolism, glycogen, it gets converted into a fuel source for creation of ATP. And also something called myoglobin, which stores oxygen. And that would make sense because, we want to take glucose, and in the environment of oxygen, we want to create vast amounts of ATP, which can result in muscle contraction. The other thing that we see that's a bit different in muscle cells because of their actions, the nuclei and the mitochondria are peripheral rather than in the middle of the cell. And I'll ask you a question. Why do you think that the body or the cells would have their nuclei and mitochondria on the peripheral aspects of the cell? The reason for that is, of course, because the cell is shortening and getting smaller, we wouldn't want the nuclei and the mitochondria within the workings of the muscle. They would get damaged, and of course, we don't want that. So here we can see the sarcolemma here surrounding the cell, right? And we can see the nucleus here on the outer edges of it. You can also see the sarcolemma pulled away here, and so that you can see underneath what's happening, right? So, remember, this is the cell membrane of this specific muscle cell. Within the sarcolemma, this cell membrane, you have small invaginations. So think of small pitted holes within the sarcolemma. So these small invaginations, and these are known as transverse tubules. And these small holes that we see in the membrane, they are holes that head deep into and around the deep fibers. So I prefer this diagram here. You can see that the sarcolemma has been pulled away. And if we look here on the right, we see these tiny little holes here, right? Again, so these are the T tubules. So these are the openings into the T tubules that we can see here along the sarcolemma. And if we look over here to the left, the sarcolemma has been pulled back, and we're seeing the inside of the sarcolemma, and we can see these tubes heading deep down into the workings of the cell, of the myofiber. And on the other side of the sarcolemma, we would see these openings that run these tubes deeply down into the cell itself. So just a reminder that, that does go down deep, because that's going to play a significant role when we talk about muscle physiology. And you can see here they've been cut away at this part of the picture. So again, on the other side of the sarcolemma, we would see these T-tubules traveling deep down, and you can see how they attach here. Why are they there? Well, they're filled with interstitial fluid that exists outside the membrane and inside the membrane. The whole role of a T-tubule is to allow the action potential, that is the stimulation from the nervous system. That stimulation is called an action potential, potential for action, and it allows it to travel along the sarcolemma. And then as it comes to the T-tubule, that action potential travels down the parts of it, travel down the T-tubule into the cell very quickly, which allows it to get very quickly into the workings of a muscle fiber so that you get a contraction. In other words, these T-tubules work so well that by allowing the action potential to go deep into the cell, you almost get an instantaneous reaction of the muscle fiber when it's stimulated by the nervous system. So again, the action potential travels along the muscle fiber or membrane, muscle cell membrane, the sarcolemma. It travels along that membrane, and wherever it hits the hole for a T-tubule, part of that action potential travels down the T-tubule to affect these very deeper workings of the muscle fiber itself. Understand, though, that when you look at an action potential, it is very much similar to the ripples of a pond when you drop a stone in the water. That ripple spreads outwards in all directions from where the stone entered the water. The same thing happens with the action potential that's created by the nervous system, is that from the point where the nerve, where it associates to the muscle fiber, where that occurs, there is an outward ripple along the membrane of the myofiber in all directions, and wherever it hits one of these openings, parts of that action potential travel down the T-tubule as the ripple continues forward. And everywhere it travels down, it's going to stimulate these inner workings that results in a contraction. We are going to talk a lot more about that when we have class next week in terms of muscle physiology. So when we think about this sarcolemma, these T-tubules that we see here, we can see that there's some inner workings here that I've related to, but we need to now start to look at them a little bit deeper. So I have mentioned this many times, that there's an interaction between two fibers or filaments when we look at muscle contraction. And these are known as myofibrils, and they are the contractile elements of the muscle cell. So the muscle cell is a myofiber, and these small thin filaments, as compared to the muscle fiber itself, so they're smaller, therefore they're called myofibrils. So the myofiber runs parallel to each other along its relationship to the other myofibers within the cell. And of course, we have the fascicles and so forth, and everything runs parallel along. These myofibrils, they also run parallel to each other along that line. So we say they are lined up parallel to each other. So these myofibrils run parallel to each other, and they too can extend the entire length of the muscle fiber. So remember when I drew pictures on the board, I would draw the white lines that would represent the connective tissue, and then I would make red parts in between. Think of that's what's happening here. So we've got the myofiber, and within the myofiber, we have contractile filaments, so they're called myofibrils, and they too run parallel to each other and extend the entire length of the muscle fiber. Within this, we have two types of protein filaments, which are microfilaments. We have a thick filament called myosin and a thin filament called actin. So we can see here in this diagram, the thin filaments are expressed by this particular color here as a small line. So this is the actin. And then we have these thick filaments here, expressed much wider, and these are myosin. It is the interaction of actin and myosin that results in muscle contraction. So we say that the relationship between these two microfilaments results in the shortening of the muscle, which we call contraction, but they're also involved with creating the cleavage furrow and cellular division in other cells. So actin and myosin do exist and play different roles in other physiological processes, such as this cellular division and the cleavage furrow. But when we look at muscle tissue, they're much more highly developed and specialized in order to have contraction occurring. The thing I want you to take away here is that ultimately a muscle contraction is shortening of the myofiber. And the shortening of the myofiber, of course, results overall in shortening of the muscle itself, which of course creates the tension in the tendons, which causes the bones to move. These myofilaments are repeated in structural contractile units. So think of things running in a unit down the entire length of the myofiber. These small contractile units are called sarcomeres. And sarcomeres are not the full length of the muscle fiber. They are sections of the muscle fiber that run the full length of the muscle fiber. So we can see here a sarcomere that's happening here. So I'm going to pause out of the slides here and just do a little bit of a drawing here so we can understand a bit better. So I'll draw them out here so we can see that, if we have a muscle fiber that's running its length, and we're saying that it runs from one attachment to the other, the entire length, and in some cases up to 30 centimeters, if we look down at a particular fiber, it is broken down into sections that run that length. And each of these sections is called a sarcomere. And it is all of these sections shortening that culminate in the entire shortening of the muscle. So the muscle fiber can run 30 centimeters, and within that 30 centimeter length of the muscle fiber, it is divided into working sections called sarcomeres. Okay, we'll come back to that shortly. So these sarcomeres then, as we can see here, right, we can see a sarcomere running from here to here, and these are all the workings. Again, in this drawing, we can see that the thick filaments are represented by these blue and purple configuration here, where my cursor is, and then the thin filaments are out here on either side. And you can see they're intimately related to each other in a parallel configuration, and it's the interaction of these two filaments, again, that result in contraction. So, when we look at these repeated sarcomeres under the microscope, what we see is that we can see repeating series of light and dark bands within the muscle cell itself. These repeated series of light and dark bands are called striations. And in an intact muscle fiber, we call these areas by different names. So the dark bands are called A bands, and the light bands are called I bands. So we can see the A band here and the I band up here light. So we've got another picture here. We can see the A band in this area, which is dark, and it is bordered by two I bands on either side, which is light. Okay, again, dark bands that are called A bands and light bands that are called I bands. Because these two bands are nearly perfectly aligned with each other, and we see these alterations of light and dark and light and dark and light and dark, this is what gives us a striated appearance. Now within each A band is a lighter region called the H zone. So we can see that there's a light area here, right? Again, I will repeat, each A band has a lighter region called the H zone. So we can see in this area here, this represents, so remember our A band is here, dark, and there's a light area in the middle of the A band, and this is known as the H zone. Each H zone, is bisected vertically by a dark line called the M line. So we can see the M line here that's dividing the H zone. So the A band is the dark zone. There's a little bit of a lighter area in the middle called the H zone. And the H zone is divided in the middle by a dark line called the M band. You can see it here as well. A band, dark. H zone a bit lighter, and a dark line called the M line, and we can see it here as well. Okay. Each H zone, as I said, is bisected vertically by a dark line called the M line. In the I band, there is a midline interruption called the Z disc, and the reason it's called the Z disc or the Z line is because it zigzags, and you can see it here zigzagging. We can also see it here in this diagram. Here's our Z disc here, and it's zigzagging across. So A band is the dark. H zone is a zone within the dark A band that's a little bit lighter. And bisecting through the H zone is the M line. When we go to the I band here, we can see it is divided down the middle by this zigzag configuration called the Z disc. And we can see it when we look at it more microscopically. Here is our A band here from here to here. Here is our M line here running down the middle, and all of this would represent the A band with the light area H zone in here and the M line dividing it. And then on this side we're in the I band, the light area, and it is again bisected by the Z disc that we can see running here. So this sarcomere is the smallest unit capable of contraction. In other words, it's the smallest contractile unit. Therefore, it is said to be the functional unit of skeletal muscle. So when we go back here to the drawing, we can say that each of these areas are the functional units of a muscle. So they break the muscle fiber down into small functional units, okay? So again, smallest unit capable of contraction. It is the region between the two successive Z discs. So if we go back here again, a sarcomere runs from Z disc to Z disc. So this area right here represents a single sarcomere. We'll look at this diagram as well. Z line or Z disc to Z disc represents the working sarcomere. All right, so it's the area of work and contraction. So we can say then the sarcomere runs between two successive Z discs, and it contains an A band flanked by half of an I band at each end. So we can see again it's in the A band and half of an A band here, all of an A band, and half of an I band represents a sarcomere. Full A band, half of an I band, represents a sarcomere running from Z disc to Z disc. It is within these sarcomeres that we see the arrangement of the actin and the myosin. The central thick filament contains myosin, which extends the entire length of the A band. So again, we can see here, this is the A band that we see here, and the thick filament, the myosin, which is represented here in brown, is where we find the A band. So the A band in a relaxed muscle state runs from the tip of one myosin to the tip of the other myosin, and they are contained within the two Z discs. Therefore, this represents a sarcomere with the myosin running within the A band. Lateral to the thick filaments, we find the thin filaments, which contain actin that extend across the I band and partway into the A band. So again, we can see here is our A band here, the dark band, and we can see half of an I band on either side, Z disc to Z disc, half of an I band. And we can say that the actin runs in the I band and partially into the A band. Okay, in the I band and partially into the A band. I'll go back again and say lateral to the thick filaments, we find the thin filaments, which contain actin that extend across the I band and partway into the A band. It is the Z disc that anchors the actin. As you can see here, the M line anchors the myosin and the Z disc anchors the actin. Again, the Z disc anchors the thin filaments. There is a third type of microfilament here, and it is called the titin or elastic filament, which is composed of a gigantic protein called titin. We can see it here in this curlicue configuration here, that it's running again from the Z disc to the tip of the myosin fiber. This elastic filament extends from the Z disc to the thick filament and then runs within the thick filament to attach to the M line. So again, we can see here it's anchored here to the Z disc. It curlicues to the tip of the myosin, and then it runs the entire inner part of the myosin to anchor right here on the M line. Right? It's not curlicued in here, but it is here within the I band between the Z disc and before you get to the tip of the myosin. So the elastic filament extends from the Z disc to the thick filament and then runs within the thick filament to attach to the M line. It helps hold the thick filament in place and helps the cell to spring back into shape after being stretched. So when we look at this and when we're stretching a muscle, all right, and what we're doing is we're having the Z discs get farther apart when we stretch. Z discs get farther apart. Therefore, this curlicue configuration of the titin molecule, it will start to get straighter and straighter and straighter until a point where it can't straighten any longer. And this is then where we start stretching the fascia that surrounds this configuration. But when the muscle relaxes, this straightened out titin, because it's springy or elastic in nature, it pulls the Z discs back to their resting position and returns back to this curlicue configuration. So we say it holds the thick filament in place and helps the cell to spring back into shape after being stretched. So as I just stated, the titin stiffens as it's stretched or uncoiled, which helps the muscle to avoid excessive stretching. Now, when we go back to the zones, we say the H zone of the A band appears less dense because the thin filaments do not extend into this region. So we go back to this picture again, we can see that the A band here has this region that's a bit different because the thin filament doesn't extend all the way to the M line. So we say the H zone of the A band, which again is this central area here, running from tip of actin to tip of actin, that represents the H zone. We say that it appears less dense because the thin filaments do not extend into this region. The M line in the center of the H zone is slightly darker because it has the presence of a 5' protein strands which anchor the thin filaments together. We can say that the microfilaments are held in the sarcomere at the Z discs and the M lines. The Z discs is where the thin filaments anchor, and the thick filaments, the myosin, anchors at the M line. So again, we've got our myosin anchoring somewhat here in the M line, also providing some anchoring because of the titin molecule. And then we have our actin molecules or filaments anchoring into the Z discs here. So thin into the Z discs, myosin or thick anchoring into the M line. Now, when we look at the relationship of these, right, we can see in this type of drawing that they're very intimately parallel to each other. And we know, as I've said, that they do have a relationship to each other which results in contraction. And you can see from a three-dimensional sense here that there is a relationship three-dimensionally, because all we're seeing here is two-dimensional views. And you can see somewhat that there's a three-dimensional look here. We can see here on the right this particular pattern. So if we look at the thick myosin, we can see that they're surrounded by thin actin. So if we tease out just this one here, we can see 1, 2, 3, 4, 5, 6. 6 actin molecules surround a single myosin. Again, if we go to the myosin beside it, 1, 2, 3, 4, 5, 6 surrounding that one. So we can see that there is a vast amount of availability of attachments of actin to myosin when we have contraction. So we say that each thick filament is surrounded by a hexagonal arrangement of 6 thin filaments, and each thin filament is enclosed by 3 thicks. So again, we can see here that they all have this very tight arrangement. Now, when we are looking at the relationship to each other, we can see that out here in the I band, there's no myosin. We just see this configuration thin. We can see here in the H zone, again, there's no actin within the myosin, so we can see the myosin here. And we can see how they relate to each other here within this hexagonal shape. And then where we have crossover between actin and myosin, we can see how this relationship works, which allows for a vast amount of connections between the actin and myosin, which is what gives us strong contractions. So we've got these, we can see here we've got these configurations of the sarcomeres in these drawings. We can see Z disc to Z disc represents a sarcomere, and we can see our M line here in the center, our thick filaments here, our thin filaments there. Now in the diagram here you can see that these sarcomeres are surrounded by this tubular configuration on its surface. Here's our sarcolemma here on the outside. We can see here are holes for our T tubules that travel deep in. And if we travel deep in, we can see that all of these sarcomeres are surrounded by a tubular network. You can see it over here as well. Here's our sarcolemma with our holes into our T tubules, remembering that it is these T tubules that allow the action potential to travel deep into the cells, or the structural sarcomeres within the muscle cell. And we can see this tubular configuration here that surrounds the actin and myosin. So within this tubular system, this is known in muscle cells as the sarcoplasmic reticulum, right? And within there, it is a fluid-filled membranous sac, and it encircles each myofibril, and it is similar to the endoplasmic reticulum of other cells. So this tubular network that surrounds the sarcomeres is called the sarcoplasmic reticulum. And within it, you find fluid. At the ends of these, they're dilated, and these are known as terminal cisternae, and these butt against the T tubule. So our terminal cisternae are this thickened area here that lie very close to the T tubule. So the sarcoplasmic reticulum represents this tubular network that we see here, and it ends in this terminal cisternae that we see right here. Again, tubular in nature and full of fluid. We can see it here as well in this drawing. This tubular network here represents the sarcoplasmic reticulum that we can see here. And then at its ends, it ends in what are called the terminal cisternae of the sarcoplasmic reticulum. And we can see that, at the Z disc of each sarcomere, we have this configuration of a terminal cisternae, then a T tubule, then a terminal cisternae of the next sarcoplasmic reticulum. We're seeing one full sarcoplasmic reticulum here that ends in its terminal cisternae, and we can see the terminal cisternae here of the next sarcoplasmic reticulum that would surround the next set of sarcomeres, and divided between the two terminal cisternae, we have this tube of the T tubule that's running all the way down, separating each of the terminal cisternae. So we say here, the dilated end sacs of the sarcoplasmic reticulum are called the terminal cisternae, and these butt against the T tubules from both sides. We look at this configuration and we can see a T tubule, a terminal cisternae, a T tubule, and a terminal cisternae. The configuration of a terminal cisternae, a T tubule, and a terminal cisternae is called a triad. So we can see it over here, terminal cisternae, and T tubule. This represents a triad. So the T tubule and the two terminal cisternae are referred to as a triad. The whole reason for the the tubular configuration of the sarcoplasmic reticulum is because its responsibility, it is fluid filled and it stores vast amounts of calcium ions. Really important to take away. The sarcoplasmic reticulum contains vast amounts of calcium ions. It is the release of calcium ions out of the sarcoplasmic reticulum that causes a muscle contraction. Without the sarcoplasmic reticulum and associated calcium, we would never have a muscle contraction. It is the fact that the sarcoplasmic reticulum stores calcium and releases that calcium that we get the interaction of the actin and myosin, which results in shortening of the sarcomere, which results in shortening of the entire muscle that we call contraction. We are going to be talking a whole lot more about that when we look at the physiology of contraction. I'm sure you guys will have some questions for me next week. I'll gladly take you up on them, which means I'll probably have the quiz next week a little bit later, which may give you an opportunity to have some questions answered. When we look at these muscle fibers, we said that within them we find stored myoglobin. And this myoglobin is a red pigment, and its whole role is to bind oxygen. So because we have myoglobin in muscle, this allows oxygen to be bound to it, which in sense means that the myoglobin is actually storing oxygen for the muscles. And we have to remember, when we looked at cellular metabolism in A&P, that, oxygen results in creating much larger amounts of ATP. So, of course, we would want a lot of myoglobin in muscle, which means that the muscle stores vast amounts of oxygen, which means that in that type of metabolism, the muscle is then able to create vast amounts of ATP. Now, when we look at the percentage of myoglobin a particular muscle cell has, there are variations of stored myoglobin and therefore oxygen, depending on what that muscle is better at or what it's used for. And the prime example we see of high and low myoglobin is when we compare chicken legs to chicken breasts. We call legs, both in turkey and chicken, we call that dark meat, and we call breast meat white meat. The reason for that is that chicken legs contain vast amounts of myoglobin, which has them look dark when they're cooked, and therefore they're red, high amounts of myoglobin. Chicken breasts don't contain high amounts of myoglobin, and therefore it looks white. If we extrapolate that out, then we can now say that obviously the legs are for more oxygen metabolism to create ATP, and the white is non-oxygen forms of ATP. So we could say that the chicken legs are aerobic in nature and that the chicken breasts are anaerobic in nature. So a lot more ATP is created anaerobically in the breast, and a lot more aerobic ATP is created in the chicken legs. So keep that in mind, that again, when we look at human muscles, some muscles contain higher amounts of myoglobin as compared to others. Also, within the variation of skeletal muscle fibers, we find that there are some muscle fibers that contract very quickly and others that contract very slowly. So we say skeletal fibers also contract at different speeds and vary in which metabolic reactions they use to generate fuel and how quickly they fatigue. So with contraction speed considering and amounts of myoglobin, then we can say that there, between the two of them, there are various ways that muscles contract fibers. So we're going to break them down into their names. The first type of muscle fiber is called slow oxidative fibers, also known as SO fibers. Under the microscope, they appear dark red, and that means that they contain vast amounts of myoglobin and lots of capillaries. And obviously, we need lots of blood supply because that's what brings oxygen to the table that the myoglobin can hang on to and store. So they appear dark, which means they have large amounts of myoglobin and capillaries. These slow oxidative fibers also contain large amounts of mitochondria, which means they form vast amounts of ATP, which of course is created mainly by aerobic respiration, because lots of capillaries and lots of myoglobin, which holds oxygen. These are considered to be slow fibers because of the slow speed in which the ATP is broken down. So these type of muscle fibers take a longer period of time to reach peak tension, and longer to reach peak tension, but these muscles are really good at sustaining contractions for long periods of time. And because of this aerobic metabolism, there's lots of ATP, so they can prolong their contractions and can sustain contractions for very, very long periods of time. The next type of fiber is called fast oxidative glycolytic fibers, also known as FOG fibers. These also contain large amounts of myoglobin in many capillaries, which means obviously there's a lot of aerobic metabolism occurring here. They too have a dark red appearance because of this excessive myoglobin, and they generate large amounts of ATP through aerobic respiration as well. The difference between fast oxidative and slow oxidative is that there's also vast amounts of glycogen here. So not only do they have lots of myoglobin, they also have lots of glycogen. And if you remember, again in A&P, I talked a little bit about how glycogen is sort of a muscle sugar. Because of this configuration, these muscle fibers are 3 to 5 times faster in breaking down ATP as compared to slow oxidative. So this equals faster contractions. So these fast oxidative glycolytic fibers are used quite a lot in sprinting and walking. Okay, so a little more anaerobic metabolism within these fibers. The next are called fast glycolytic fibers, also known as FG fibers. They have low amounts of myoglobin and much less capillaries and mitochondria as compared to the previous two. And when we look at these under the microscope, because of this lack of myoglobin, they appear white in color. They also contain a very large amount of glycogen, which is muscle sugar. And of course, we have to think about glucose, right? That it's created in the whole process before we even get into the Krebs cycle and so forth. So these fibers, because of the lack of capillaries and myoglobin, they generally create ATP through glycolysis or anaerobic metabolism. These contract strongly and quickly, but they cannot sustain contraction. So we say they contract strongly and quickly, and they fatigue quickly. And these are the muscles we use when we're weightlifting and throwing things. When we train these fibers, we get increased glycogen stores and increased size. So this is, weightlifting, speed work, that kind of stuff. So training these fast glycolytic fibers results in increased glycogen and increased size. So how do we look at the distribution of these three particular fibers? So we're going to look at slow oxidative and fast oxidative first. Typically, skeletal muscles contain all three types, even though we've separated them out here. Half of the fibers in a typical skeletal muscle are generally slow oxidative. Now, depending on the role of a particular muscle, the proportions of the three fibers really depends on whatever that muscle is used for. For example, these postural muscles that we find in the back, neck, and legs, the ones that hold us up when we're sitting or standing, they have a very high percentage of slow oxidative because they have to sustain muscle contractions for very long periods of time that gives us our postural positions. Muscles of the shoulders and the arms have a high percentage of fast glycolytic, they're the throwing muscles, for example. And leg muscles used for walking and running have large numbers of both slow oxidative and fast oxidative glycolytic, and therefore, long and short periods of muscle contraction. The chicken would be no different, right? Shoulders, arms, chest, white, fast glycolytic, leg muscles, walking, running, large amounts of slow oxidative and fast oxidative glycolytic, so glycogen and lots of anaerobic work, therefore white, and we see what happens there. Thanks for listening to the slide set. There's a couple more yet to come. Thanks again. Bye for now.